Good evening, everyone, and a warm welcome. I'm Professor Dylan Edwards. I'm the Provost Chancellor for the Faculty of Medicine and Health Sciences at UEA, and it's a huge pleasure tonight to be introducing Professor Ailsa Welch. Ailsa is a Professor of Nutritional Epidemiology based in the Norwich Medical School. She holds a degree in nutrition and spent 15 years as a Senior Research Associate in Nutrition at the Department of Public Health and Primary Care at the University of Cambridge. And it was during that time that she developed dietary assessment methodologies for the European Prospective Intervention and Cancer, EPIC, the EPIC study, which is actually, interestingly, it's the Norwich population. Some of you may be involved, maybe subjects of that population study. So, important stuff. She has also worked as an information scientist at the Royal Society of Chemistry and was at King's College London working on projects based in Somalia and Nigeria. I didn't know that about you, actually. She also obtained her doctorate from the University of Ulster during this time. Ailsa joined UEA in 2007 as a lecturer in nutritional epidemiology in the medical school. Her research focuses on understanding the protective factors in diet for musculoskeletal health, osteoporosis, and fracture risk. She also researches cardiovascular disease and cancer, amongst other things, in developing dietary assessment methodologies, quantifying measurement error, and in evaluating public health improvement interventions. Ailsa's research aims to understand the effects of micronutrients, that's things like vitamins and minerals, and diet quality on musculoskeletal and cardiovascular health. Understanding how diet improves health during aging is important, and we can see that from the audience tonight as well, as the age profile of populations is increasing with consequences for the burden of health on health and society, social care. Micronutrient malnutrition also coexists alongside obesity and chronic disease. Ailsa has been also a state-registered dietitian since 1979. I didn't know that about you either as well. And has also advised the UK government in relation to dietary surveys. Ailsa chairs the Nutrition and Lifestyle Forum of the National Osteoporosis Society and is a member of their Scientific Advisory Committee. She's also the scientific theme lead for public health nutrition for the UK Nutrition Society and chairs the initiative for optimising nutrition and hydration practice in care homes. She's an external examiner for the London School of Hygiene and Tropical Medicine. Ailsa is also one of our most accomplished scientists. She's a top 1% world researcher in the Thomson Reuters 2014 rankings and has 197 peer-reviewed publications and 14,666 citations and counting. So there you go. We're going to cite a few more after tonight. So please join me now in welcoming Professor Ailsa Welch to give her inaugural lecture.
So thank you very much, Lynn, for that very nice introduction. And I'd also like to thank everyone for coming here tonight, because there's obviously a lot of people here. And I know some of you have come from a very long way, some from as far as Surrey and Cambridge and Saffron Walden. So it's very nice for you to be here, particularly on this sort of cold and dark night. Okay, so it's said that there are two certainties in life, death and taxes. And I think we might argue, you might argue your way out of taxes, but you certainly won't argue your way out of the other one. But the third inevitability is that we're all getting older, and every year we increase in age. There's no denying that, and that isn't going to change anything. But recent evidence from myself and from a number of many other researchers across the world is finding that nutrition impacts on the aging process. So what I've been using is the technique of nutrition epidemiology, and this means studying people in populations to understand the differences across those populations, to try and find out what would be protective for preventing the onset of chronic diseases and aging. And by chronic diseases, I mean diseases such as cardiovascular disease and diabetes. And also I'm going to be talking later about other aspects of health and aging. But diet, when we study diet, we can think about the whole diet in terms of dietary patterns. We can think about individual foods, and we can also think about the foods within those. So as Dylan said, a large part of my career has been spent measuring dietary intake and trying to understand how we can do that better. Developing dietary assessment methods, but also developing and understanding other measures of nutrition using nutritional biomarkers, and that means measures of nutrition that we can make either in blood or in urine and other tissues, and I'll talk a little bit about that later. And the reason for doing this is really to try and measure what we do much more accurately and without what we call measurement error. Whenever we do anything in terms of science, we're always going to have some amount of error, and the idea is to try and change that and minimize that so we get more accurate results from studies. So perhaps longer ago, I published in relation to cancer and diet, but also more recently still publishing in relation to cardiovascular disease and risk of stroke. And recently you may have seen a paper that I published where we looked at the relationship between the Mediterranean diet and risk of stroke, and that was in EPIC Norfolk participants here. But I'm not talking about that so much today. But also I wanted to note that I've had a long-term association and collaboration with public health colleagues in Norfolk, particularly in the County Council more recently, where we've been evaluating health improvement initiatives. And that's helped to decide whether people continue to fund the initiatives and to know whether they're effective or not, because a lot of work goes on and we often don't know whether it's actually really effective. So today, though, I'm going to focus on nutrition, aging of muscle and bones. And I'm particularly going to focus on muscle because this is an area that there's been much less research on, it's much more recent research, and it's an area which you may not have encountered quite so much. And I'll be talking about micronutrients in particular, and these micronutrients are basically vitamins and minerals. So we know there are a number of effects of aging. We know that we have the baby boomers, many baby boomers in the population, and they are increasing the bulge in age in the population as we go through. And this is causing an issue for governments, and also it's contributing to issues that we have for how we cope in health, in social care, in the hospitals, and in other areas such as care homes and social care. So this is putting pressure on everybody, and we hear, almost daily there are a number of headlines about this. So what we really need to be doing is keeping the population in good health and keeping them mobile, so keeping everybody moving around, rather than needing to start to become disabled and needing a lot of help from a lot of people in society. So, and we need to also try then to reduce health service requirements. So I mean, this is the sort of global picture, but actually also for ourselves, we need to know as individuals what we're going to do to maintain our health, particularly of our muscles and bones, and I'll tell you why I think that's important later. And also for then how we can think about how that would impact on policy. So there's 4 parts to my talk today. The first, I'm going to talk about sarcopenia, which I'm going to explain a little bit more about in a minute, and osteoporosis and fractures. And then I'll move on to talk about diet and mechanisms of aging of skeletal muscle and bones. Then thinking about nutrition epidemiology, particularly about cohorts, and then providing you some more recent results, some of which are published and some of which are in the process of being published. And lastly, I'm going to talk about the implications of micronutrient malnutrition. So many of you will know about osteoporosis. You'll be familiar with this term, and you'll know that the osteoporosis is low bone density. And this graphic here shows you a graphic of normal bones. This is young bone, which is well structured, and osteoporotic bone, which has lost a very large part of its structure. And this is why it becomes weak and people are more prone to fractures. You're less likely to have heard of sarcopenia, although many of you will, because it's starting to be recognized. We've known for a long time that there's something going on in people, and we've recognized it in clinical practice and so on, but we didn't really have good definitions to understand what this was and how to really characterize it. And so we've been, but now we know that it's low skeletal muscle mass and function. Sorry, I've lost my cursor here. And this definition really only came at around 2010, and it's been revised. Because this is such a young and growing field, it's a very interesting area of science, but we haven't quite completed how we would define sarcopenia. So, it's a case of watch this space, but certainly there's a lot of work ongoing. So what's going on in sarcopenia? This is showing you some muscle mass in 2 people. So muscle, a cross-sectional area of muscle in a young person and an older woman. And you can see they've got a similar volume of muscle, of leg area, but what you can see in the younger woman, this is much more dense, and in the older woman, there's a lot more fat, and there's a lot more fat infiltration. And what's also going on with this is that actually the fibers are changing, so you're moving from the fast twitch fibers to the slow twitch fibers as well. There's a number, many activities going on here. So in this talk I'm going to talk, we generally measure skeletal muscle as fat-free mass, but I'll also be talking, but in general I'm going to be calling it muscle in terms of shorthand. You may or may not know too is that men and women have different amounts of muscle in their body, and they always do from very early on in life. So women of around the age of 50, 60 have around 60% muscle, and men have nearer 80%, and that's just differences in biology. So there are other factors that mean that we should be thinking about how muscle and bone interact and the links between them. One of them is that actually the structure of both these tissues involves collagen, and collagen is involved very much in the structure of bone. It's also part of the structure of muscle and also of tendons. But other things that we've started to realize more recently, I think we've very often been, as scientists, been thinking about bone, or we've been thinking about muscle. We haven't necessarily thought about them together. But what we know now is that many of the risk factors that cause either low bone or low muscle mass link together, so for both bone and muscle. So age is one risk factor, smoking another one, low weight-bearing activity or low physical activity, and extreme alcohol consumption. They're all very strong factors for losing both muscle and bone. And the mechanisms of aging are also very similar, or some of them anyway, and I'm going to talk in more detail about that in a bit. So in terms of sarcopenia, I've told you that it's low muscle mass and function. You may be less also familiar with the term sarcopenic obesity. So this is basically when somebody has sarcopenia and they're also overweight or obese. It means they're weak, and you might not recognize them in the same way as someone who might look frail and a little bit unable to cope with life. You may not actually realize that they might look quite robust, but they still will have difficulties and weakness. So sarcopenia, we have an idea now of how prevalent it is in the population. In people over the age of 60, I think around about a third of people have sarcopenia, and up to at least a third of people who are in care homes and long-term care. Frailty, which is another condition that is sarcopenia contributes to, is present in around about a quarter of people over the age of 80. So we know about weakness and we know about muscle loss in sarcopenia. What people have really focused on much less, certainly in nutrition and science generally, is muscle is a very important metabolic organ. So essentially what it is the main organ that metabolizes our nutrients and our energy. Adipose tissue is relatively inert compared with muscle. So if we lose muscle, we're starting to lose energy-burning tissue. We also know that muscle is the main destination of glucose. So we have glucose circulating in the blood, it goes into muscle. Now if we start to lose, so it makes sense, if we start to lose that volume of muscle, we're going to have less ability to metabolize those nutrients. And I'm just showing you here energy and glucose, but there are other nutrients too that are impacted. So this can be impacting on the development and onset of other chronic diseases, particularly type 2 diabetes and obesity. And this also contributes to the onset of frailty. Sorry about the noise, it keeps making that noise when I misclick something. So, I mean, the reason I became interested in muscle was particularly because I was working on osteoporosis and on falls and fractures, and realized that actually muscle was very important, because if you can prevent falls, you can technically prevent fractures. And clearly we want to be preventing fractures. I mean, there are many reasons for this, but one of them is that by the time we reach 50, 1 in 2 men and 1 in 5 1 in 2 women, and 1 in 5 men will sustain a fracture. So that means any of you sitting in a row, I mean, that's 1 in 2, it sounds quite a lot, but actually any of you sitting in a row along here, if you're sitting in a row next or somewhere similar to somebody who's over the age of 50, either you or the person next to you in that row is going to have a fracture, an osteoporotic fracture at some point in the future. And it doesn't not just affect, men are also impacted. Now, the other problem with having a fracture is statistics are really difficult and so on, but actually if you have a fracture, particularly a vertebral fracture, it's painful and takes a long time to recover from. And also know that if someone has a hip fracture, around about half the people who have a hip fracture actually are unable to walk unaided after a year. So this is kind of causing, many problems obviously to governments and so on, and the costs of all these conditions are huge, billions of pounds a year, but also it's costing the human cost is actually really very troublesome. So, we recognise frailty, we recognise people are old, but actually what you may or may not realise is that ageing is happening very slowly and it's continuous after the age of 30. And this graphic shows you what's happening with muscle and muscle strength. So what we see is that overall there's a build-up of muscle strength in early life until around about the age of 30. This is in childhood and adolescence and so on. It's obviously very clearly very important that people live in the right conditions and have the right nutrition in order to attain this. But then gradually through adult life there are reductions. So this graphic shows very nicely the range of the reduction. It might decline slowly or might decline fast, and you see that there would be a difference. And for strength you can see that this comes through to a threshold where physical performance is impaired. So this graph actually, a fact, shows what's happening in many body compartments and systems. So we know this bone density reflects this, skeletal muscle mass is the same, and sex hormones too also decline very slowly. And we don't notice at this point in the midlife, we tend not to notice, although we might notice we're getting weaker. So this progressive decline does tend to steepen after around the age of 50, and we know there are gender differences between men and women because men decline much faster than women, but women particularly also decline the menopause. So I'm going to give you a couple of figures here for how much this decline is, and I'll ask you to try and remember this for when I come to some of the later results that I'm going to show you. So what we know is that muscle mass declines between about 1 to 2% a year after the age of 50, and force and strength between 1 to 3%. So you might think, well, that's actually not very much really, maybe that doesn't matter totally. But actually, if you're sitting here at the age of 50, by the time in the next 10 years, you could have lost 10% of your muscle mass, which starts to sound like actually quite a lot. So what we want to be doing is really to prevent these declines and to promote a healthy lifespan and slow the progress to sarcopenia and frailty. So what can we do to stop the condition? Well, in terms of sarcopenia, we know that clearly having low physical activity is a risk factor, but what we know is that even athletes lose muscle mass and function as we age. There is research on diet, but largely that's been done in the past on young athletes and men and on sports performance, comparatively little on ageing. Protein has been studied, and we know that we do need protein. We have to maintain the balance between protein build-up and protein breakdown. So this is this figure here showing the anabolism and the catabolism. And what we know also is that interventions in older people have very mixed success with protein. Sometimes they're successful and sometimes they're not, and they do tend to be more successful if they also include resistance activity. But it struck me around sort of the about 2008, 2009, I couldn't actually work out why people hadn't been investigating other aspects of diet in terms of muscle aging. Didn't really know why, and to be honest, I still don't really know why. But I started to look into this with PhD students and research associates and a number of other people here. And I'll show you some of the results in a few minutes. But so what I've already published is and shown that the fat quality of the diet does have an impact on how much muscle we have. Also, how alkaline the diet is has an impact on that. And also, I've published on dietary patterns such as the Mediterranean diet. But there's been much less work also on micronutrient vitamins and minerals, and I'm going to show you more data today on magnesium, vitamin C, and carotenoids. And whilst we know that patterns are interesting and they're very useful, one of the reasons I'm interested in what's going on with these micronutrients is because essentially the patterns are working through the content of the micronutrients in them, and also the macronutrients, but very much the micronutrients, that's the vitamins and minerals. Okay, so now I'm going to move on to a section thinking about how diet influences the mechanisms of aging of muscle and bones. And you'll all be aware that calcium is good for bones. I think everyone knows that. But you may be less aware that calcium is important also for muscle function. We need sufficient calcium in order for muscles to contract. And we know, I've told you about protein, and we know that that also is important for muscle, but also it's important for the structure of bone, and particularly in relation to collagen, which I mentioned earlier. Other nutrients we know are important for bone are vitamin D, which we can get from diet, and we also get from exposure to the sunshine. And I've already talked a little about dietary patterns and muscle, but today I'm going to talk much more about these nutrients that I mentioned earlier, vitamin C, carotenoids and magnesium. So this graphic shows some selective mechanisms for how muscle and bone are aging. So one of the things that happens is that when we metabolize, basically when we're living, we're metabolizing nutrients, we're metabolizing oxygen and so on, and during that process we produce compounds called reactive oxygen species, and the short-term name for that is oxidative stress. And as we get older, more oxidative stress is occurring in our bodies due to the aging process. And this can cause damage to a number of the body compartments and a number of the different tissues in the body, including DNA. We also know that there's inflammation, or low-grade inflammatory cytokines are also increasing as we age. So these are compounds such as TNF-alpha, if you want to know more detail. But basically, these are the sort of response mechanisms to, for instance, if we have an infection, they're much more low-grade with aging, but they're continuous and they're causing damage. And there's also a protein called C-reactive protein, which is another indicator of that. So what we know is that inflammation impacts on protein turnover, and that balance that I was showing you earlier, the building and the breakdown of the protein, as does oxidative stress. But also, to maintain bone health, we need to have a balance of the sort of cells that actually build bone and break bone down. And these are the osteoblasts and osteoclasts. And these are also affected by inflammation and oxidative stress. So where does diet come in? Well, this is one of the factors that really impacts on this, is that we know that if you've got a deficiency of a nutrient that contributes to structure, that's obviously going to cause the body problems. So if you're deficient of calcium, magnesium or protein, that's going to cause big problems to the body, as is the whole area that we have here of the different mechanisms. But what we know is that whereas these are called oxidative reactive oxygen species, we know that actually we have antioxidant molecules in the body which will deal with oxidation. They're called endogenous antioxidants. We have exogenous antioxidants which will also work, and these are the vitamin C, E, carotenoids and magnesium. Okay, so I'm going to talk to you now a little bit more about a group of compounds called carotenes or carotenoids. And their major sources are from the brightly coloured fruits and vegetables, and you can see here that they are very brightly coloured. And what we have here is carrots supply a lot of a compound called alpha and beta carotene. We have other fruits, particularly brightly coloured orange fruits and citrus, provide compounds called zeaxanthin and beta-cryptoxanthin. Now, I'm not going to test you on this. I don't necessarily want you to remember it particularly. What I want you to know is that there's a whole lot of different compounds. They're very different, and they come from different types of fruits and vegetables. That's the basic message. So so it's just so that you can see. Lycopene is largely only found in tomatoes. And then lutein also comes from, we find the main sources in the UK are from peas and lettuces and green salads and broccoli and so on. So these whole sets of carotenoids, they have their own activity. Each of them are different levels of powerful antioxidants, but also when I'm showing you data on total carotenoids, this is the sum of these factors. Carotenes also are relevant for eye health, and I'm not talking about that today, but they also are converted to retinol or vitamin A, which is also an essential vitamin. Okay, so now a couple more, bit more information about the specific mechanisms of magnesium and vitamin C in particular. So magnesium is particularly interesting because it's been studied a little bit, but not a great deal in aging in terms of muscle. But what we know is it's integral to metabolism, so I've talked earlier about energy production, and ATP is one of the factors that is generated that allows muscles to move and us to live in general. We also know it's highly important. Magnesium is important for both muscle and bone because it's found in high concentrations. There's a lot of magnesium in bone and also in muscle. And in animal studies, we know that it's important also for protein synthesis and turnover. Inga Dorop some while ago did some study where she made rats deficient. What she found was that protein synthesis and turnover, that balance I was showing you earlier, was completely disturbed by magnesium deficiency. On the other hand, I think we haven't been thinking enough about vitamin C and how this contributes to collagen synthesis. I've already shown you through my talk already where collagen is found and why it's important. And what we need is vitamin C to make sure we can hydroxylate proline. So what proline is one of the amino acids which is the building block of collagen. And if we don't have enough vitamin C, we're not going to create our collagen very well and it's going to become weak and distorted. So you know, it's one of those links that we perhaps have thought less about. So the reason I'm going to show you the data in the next few slides is because there's been very little research on this area. So now I'm moving on to talking about nutrition epidemiology in terms of cohorts and results, and some historical as well as some current work. So many of you have heard about the European Prospective Investigations into Cancer and Nutrition. This was started in the late 1980s at the Agency for Research on Cancer by Elia Ribali and Nick Day. And it was a very novel idea at the time. Nick Day moved to Cambridge, and then Katie Core joined him as a principal investigator. Sheila Bingham and also joined, and I worked with her for many years on the dietary assessment method area. And then Wareham joined later, and he's now taking forward EPIC into the future. So this is a photo too of some of the other people that worked with us. Robert Lubin, who I'm not sure if he's here, I know he's planning on coming tonight, worked with Robert many years on developing databases and the whole sort of area for how we capture data, do the statistical analysis in EPIC. And Susie Oakes, who was also worked with us for many years, and then followed by Shabina Hyatt here, and they managed the data collection and so on. So when EPIC started, it was designed because you can see that it covers a large part of Europe. This is 10 countries that took part. We've got all the way from the south up to the north, and in fact they had to redraw this map to include Tromsø because it's so far north. And but one of the reasons it was started was because we wanted to investigate cancer, but what we knew was that diet varied a lot, but actually the rates of cancer did not vary so much. So there was something going on here that was going to be highly important. So overall, eventually, we recruited nearly 500,000 people into EPIC, and in EPIC Norfolk, we recruited 25,000 people. And I was involved in the days. We spent many hours sitting in Lyon, deciding how we were going to coordinate our methods, how we were going to make comparable information. So we compared diet, how we could collect the blood in the same way, how we could measure everything. So when we were measuring information and comparing it, we were more certain that we were finding something important scientifically. But I do recall the first days when we'd been sitting through and we got all these questionnaires and all sorts of information, sat there with empty filing cabinets, and then we sent the post out. And we waited, thought, is anyone going to reply? And they did, and the people in EPIC in Norfolk really responded extremely well, and over time, we built up the cohort of 25,000 people. So although this was designed to really think about cancer and understand diet and cancer, Nick Day and Core were very far-sighted in thinking too that we really wanted to be thinking about aging and other chronic diseases such as cardiovascular disease and diabetes. And EPIC itself has gone on to publish many, many studies on a range of different health issues in relation to in many areas. We collected information at baseline in '93, and then there were a number of follow-ups over time. But what I want to point out to you is the prospective design of the study and why it was important. We call a cross-sectional study one that looks at differences across people at the same time. Prospective means when we recruit people into a study and then we follow them up with time to find an endpoint. And this might be something like, for instance, a stroke or a heart attack, or it might be a fracture. And the reason why it's important to sort of have this prospective information is because, particularly for diet, what happens is that once you've got a condition, people often think, oh, I wonder what caused that, I wonder what happened, and it might distort the way they remember what they were really doing when they were exposed to the factor that made a difference. So it's one way of reducing the amount of error, and it gives a lot more certainty if you have information that's come from a prospective study than from a cross-sectional study. The other factor that made this a very unique study at the time was that there were cohorts in America and other places, but very few of them had stored blood, and in order to actually measure either genes or biochemistry. And actually at the time when we were collecting blood, we were thinking about measuring genes, but actually the terms such as polymorphism and SNP really had only just begun. We couldn't even imagine trying to measure that sort of amount of information in 25,000 people or even 100,000 people. The technology wasn't there, but the PIs had the foresight to think about that. What's the foresight to think about how you're going to get at blood if you want to do some analysis? Because the thing is that you can't put a lot of information, to store blood you have to keep it in freezers, and in fact we stored it in dry ice. And so the thing was they were trying to ferret around with dry ice. If you've ever looked at it, there's lots of smoke and stuff, and you've got someone to go in, they've got to defrost thing, get an amount and put it back in. Well, we knew that was going to be a no-no, particularly for the numbers of people. So what happened was we had a very dedicated team in Norfolk who actually, we created aliquots of all these different types, which means different small amounts of blood put into straws, and they were labelled up so you could see them in dry ice and so on. That was really very unique. Okay, so I spent a lot of my time measuring nutrition and developing the methods for EPIC, and this is really one of the few slides I have to just talk about that very briefly. We decided to measure nutrition using a number of different methods. One of them was using a 7-day diary, which you can see here. And what you can see is people were asked to write down lots of detail about what they ate. This person's wrote down apple pie, homemade, portion size 3B and so on. And we had 7 days worth of this for everybody, and we ended up with 25,000 of these, which is an awful lot of data. And so I developed with teams in EPIC a program called DINA, which was Data into Nutrients for Epidemiological Research. Because we needed some way of computing this and trying to analyse it. And when we finished, the data sort of is now all in for the 20,000. It took many years of a lot of people's work. We had 6 million lines of data. It's a very great deal of data. If any of you got, when you think about computing and how it used to be and how it is now, that's still a lot of data. And we had at least 200 nutrients that we developed with databases. We also, the team, we developed the European EPICsoft program, which was an electronic 24-hour recall, which we used as well. So I want to note briefly a little bit of information about nutritional biomarkers, because I'm going to be showing you some data in a minute, and just to talk again about what those are. So what this means is that you can measure nutrients in blood or adipose tissue, which is fat tissue, or you can measure it perhaps in urine, such as in 24-hour samples. The advantages are that essentially that it's independent of reporting. So somebody might tell you what they're eating, but they may not always either tell the truth, or they might be a little bit off, or whatever, we don't know. There can be some bias. And so this allows us to look at this independently. But also if we find something that relates to an outcome, which is an endpoint or something like, for instance, a fracture, those are all terms I'm using in order to explain what we'd be looking at the end in the study. This allows you to have extra confidence in the findings. Okay, so now I'm going to move on to some of the results of the research that I've been using from the study, some from EPIC Norfolk, which I've already told you something about, Biobank, which is the UK Biobank study, which is a study of 500,000 people in the UK, and you can see the distribution is throughout the country, in around about 160,000 people. I say that number, 160,000, doesn't seem very much. When you think how many people, how many numbers of this lecture theatre would have to generate this amount of data, it's huge. The number of people taking part is just enormous. And then also the UK twin study, which with just about 2,500 women. But what you'll notice is the ages are slightly different. This EPIC Norfolk was the oldest cohort in terms of recruitment. the slightly younger age group in biobank, and in the twin women, a much broader age range, so we could look at all ages. So some of the data I'm going to show you now is outcomes, we'll say outcomes or endpoints for bone and sarcopenia and muscle. We measured bone mineral density using, which is not technically that, but I'm going to call it that during the rest of this talk, using heel bone ultrasound attenuation. And here you can see one of the EPIC participants having his heel bone. We also looked at incident fractures, which means when I talked to you earlier about prospective, that they were fractures that occurred during the course of the study, and we followed people up for those. For muscle, we measured fat-free mass, which I'm going to call muscle, either using DEXA or bioelectrical impedance, and you can see somebody here having their body composition measured as well. Grip strength, we used this dynamometer to measure, and although it's a very simple measure, it does actually predict both mortality and hospital admissions. And then in the twin study, we also had a measure called leg explosive power, which I'll call power. So I've already talked to you about what muscle mass is, or fat-free mass, but one of the things that you may or may not know is that if you're taller or heavier, you have more muscle mass. And clearly across a population, across a room of this size, everybody's a different shape and size. So in order to really understand what's happening with muscle, we have to make adjustments to scale across populations. And so I'm going to show you some data where we scaled according to body weight as a fat-free mass as a percent, and the other one where we scaled for BMI. We've got dietary data for magnesium, vitamin C and carotenes, and for blood we've also got data for magnesium, vitamin C. I've divided the data into quintiles, which means you decide when people are eating, once you've got the information that people have given you, you divide it into 5 parts, the bottom 20%, the second 20%, and so on, and you compare it across those 5 categories. We also have data for circulating C-reactive protein. And so we adjusted these outcomes in statistics using age, as you would know, but also we adjusted for protein and calcium for both for bone and for muscle. And I'm showing you some data for men or for women depending on what comes through. So this is the first slide showing the relationship between magnesium intake and muscle mass and leg explosive power in twin women. And this is in all ages. So on this axis you can see this is the amount of muscle percentage, this is the bottom 20% and the top 20% of intake, and this is what they're eating, so 225 milligrams of magnesium in the bottom group and 481 in the top, so that shows us a big difference in what they're eating. I've also calculated the difference across this steep gradient here, to make sure that we can compare this across the different data I'm going to show you shortly. And there's a 4.3% difference across that. And if you think a little across back to when I was telling you about the amount of muscle you're losing every year in terms of the percentages, I said that was between 1 and 2%. And this was significant. So I'm not going to talk any more about that, but where you see this P and 001, it means that that's a very strong association. Okay, so this is data for the leg explosive power, the same idea. This is a measure of force, and you can see increasing intake shows the difference. The difference between these two was about 20 watts per kilogram. This was a massive 24% difference between the top and the bottom amount of what people were eating. That's extremely large even in epidemiological terms. So it shows that something is going on with magnesium in terms of muscle power. So when I told you earlier that magnesium is relevant for protein synthesis and it's relevant for ATP generation and so on, this is probably how it's working. And actually in the analysis what we found was the effects of magnesium were greater than for protein. I mentioned earlier C-reactive protein being one of those factors that we count as an inflammatory factor that will also potentially lead to loss of muscle. Well, our bone too, but I'm not talking about that now. So what we looked at in statistical analysis was the relationship between C-reactive protein and the amount of muscle someone had. and looked at the relationship. And then we looked to see what happened when we worked out whether magnesium would impact on that. And what we found was that magnesium reduced the association between the detrimental C-reactive protein and skeletal muscle by about 6.5%, which again, if you think back to those figures that I showed you about how much you're losing every year, that's actually quite a big effect, because this was on top of all those other factors that we adjusted for. So, those data were in women. I'm going to show you now some data for men from EPIC Norfolk. Again, a similar structure to the graphs, this time using fat-free mass adjusted for BMI. And this time also looking at different age groups. So the light blue are those over the age of 60, and these are the age of under 60, and these are over 60. And what you can see again, you've got strong trends and big differences, and the differences are between around 5%, which again is large. And we found similar but larger associations in women of 6 to 7%. We also looked at magnesium in blood, but we didn't find any association with that. And before this, we'd also looked at bone density and fractures, and we've published this data, so if you're at all interested, you can look up these publications on the website. We also looked previously at bone and fractures, and we found in relation to magnesium intake, magnesium in the blood, and also potassium intake. And in general, we found protective associations both for bone density and also for fractures of the hip and spine. And one of the last slides I'm going to show you on magnesium is data from the biobank that I told you about, looking at men and women in different age groups. This is the very much larger population, and again we're finding similar differences across the quintiles of about 5% in muscle mass and 1.1 to 2.4% in grip strength. Not so large for that. For BMD or bone, we found larger effects in men than women, and similar effects in those above and below the age of 65 years. So if you remember, I said that prospective findings are stronger than cross-sectional, and we followed up this analysis looking forward to see what happened over time, and we found that actually magnesium was protective for both muscle grip and bone strength in these findings, although we haven't yet published this. So now I'm going to move on to the different nutrient, which is vitamin C, and this is some work I've been doing with Lucy Lewis, one of the MBBS students, and shows you a similar graph to before. We've looked at both diet and blood concentrations of vitamin C. So this is data for women. Again, it's a similar sort of the structure from before, but this time I'm only showing you 3 bars, the lowest 20%, the highest 20%, and the middle. So what you can see across diet, we've got differences of about 3% in men and women. And for blood, we've looked at the difference between what would be considered an adequate amount of vitamin C in the blood and what would be considered to be a low vitamin C. And you can see that the differences there are very similar to those with diet. So what it means is that what we're measuring diet is actually a little bit more confident that it's not just what people are reporting is actually really happening with people. What I wanted to point out to you here was the amount of vitamin C people are consuming in this bottom 20%. And actually they're consuming, on average, 37 milligrams of vitamin C a day. Well, our UK recommendations for vitamin C are quite old, and they still stand, and they're developed for preventing scurvy, but the level we recommend is around 40 milligrams per day. So that means that those 20% of people are below that level. So we have more recent recommendations from the European Food Safety Authority, who recommend 80 milligrams per day. And you can see, therefore, that people in this middle quintile are also low. So what it means is that most of the people in this, about 60% of the people in EPIC Norfolk, probably had blood levels that are not necessarily as good as they could be. We've also looked previously at the relationship between bone and fractures and vitamin C and diet and blood, and finding generally protective associations. So now I'm moving lastly to carotenes in diet, and this is data that Richard Hayhoe has worked on, looking at carotenes in relation to bone density. What you can see across here, you've got some nice looking p-values. All these are significant, And you can find that in general with an increase, you're getting better levels of bone. Very big variability clearly, and the effects are quite small. So beta-carotene was the one I talked to you about that was found in carrots. These beta-cryptoxanthins are found in the orange fruits and veg, and lutein is the xanthin. These ones that we find in the peas and the lettuce and so on. Lycopene in tomatoes. So all these are proving to be predictive in women, in diet in women in relation to their bone. Retinol, which is the vitamin A, was actually going in the opposite direction. We also looked at fractures as well, and we found some mixed associations and also in blood. But in general, I think this is the first data that's been published that looks at both diet and blood and all the different fractions in relation to bone. We've also got work ongoing in the UK twins showing that actually in relation to both muscle and power, these carotenoids are actually protective, and we'll be publishing that hopefully soon. So to summarise this, what we're finding across those nutrients, these antioxidant nutrients, is that we've got difference in muscle mass of around 2 to 7% for magnesium, around about 25% for muscle mass, for power rather. We've got vitamin C muscle about 2 to 4%, carotene similar 1 to 2%, and power around 7.5%. So they're all having some positive effect on muscle. And also we found protection for fractures and bone density. But all these are accounting after the factors that we generally know about. So we know calcium is good for bone and protein good for muscle, but they're on top of that. And basically compared to those differences I showed you earlier about the gradual decline, they are of potential clinical significance. So now I'm going to take a different tack and talk about the implications of micronutrient malnutrition. So thinking about, I've talked to you about the fact that over the range of nutrients there are differences in the way in both muscle and bone. One of the things that we need to think about is what are the effects of micronutrient deficiency. Well, you'll know that vitamin C deficiency leads to scurvy, and the symptoms of this are listlessness, general malaise, weakness and fatigue, and psychomotor issues. Pre-scurvy is similar. For magnesium, we know we have weakness and muscular symptoms and so on. And for a number of the other micronutrients, which I haven't talked in detail today, anemia causes lethargy, weakness and fatigue. And vitamin D deficiency causes osteomalacia and painful muscles and fatigue. So all these are caused by low intakes of these nutrients, and particularly magnesium deficiency is also caused by clinical conditions, certain drugs, proton pump inhibitors, and also diuretics, and also higher alcohol intake. So, but actually, one of the things that should strike you is that these really are the symptoms of aging. So, we've got people who feel old and frail and tired, but how much is this down to micronutrient deficiency? I would suggest that may be quite important. So, I've told you where we get the carotenoids from, we've looked at that. Vitamin C is also coming from these foods here, and also, fortunately for us, potatoes, because a deficiency of these, not consuming these really makes a difference to how much vitamin C you have. And then magnesium comes from a much broader range of foods, including the vegetables, green vegetables, meats, fish, and also cereal foods, particularly health. So how are we doing on vitamin deficiency in this country? Well, we have some really tremendous surveys in this country which tell us what's going on with the nutritional health of the nation. So when we look at fruits and vegetables today, this is very recent data, we find that of people aged 65 to 74, only a third of those are consuming 5 portions of fruits and vegetables a day. When we go up to the age of 75, that's 20%. So actually, when I've just told you where the vitamin C is coming from, and they're not consuming fruits and vegetables, those people, the majority of these people are going to be really rather low on vitamin C. If we measure magnesium intake as well, again, this is low. It's not so bad as for fruits and vegetables, but it's about 10% in the younger age group and nearly a quarter of those in the older age group. Scurvy measurements themselves, which are less than 11, in a measure of 11 micromole per litre in blood. Free living people, 14%. Institutions such as care homes, 41%. Magnesium itself is much more difficult to measure and more difficult to interpret, but essentially around 10% of people are thought to be deficient. And when it comes to iron deficiency, vitamin D deficiency, institutions, this is around 40–50%. So this is actually very substantial. And when I talked to you about actually earlier about where the nutrients come from and the effects they're having, I think this is a real issue. So in summary, at stage, we need to think about conserving skeletal muscle mass and function in middle and early older age to prevent sarcopenia, frailty, osteoporosis and fractures. I've shown you that there is actually a very great continuum of diet, and that diet does affect how much muscle we have and how it functions, and this is important in all age groups. And clearly we need micronutrients as well as protein for preventing sarcopenia and osteoporosis. But right now we don't have any specific dietary guidelines, either muscle or bone, or in older age groups in terms of how we are telling people to eat. So what do we do? Well, the first recommendation is to, as we have no dedicated guidelines, to look at eating well in general. And this is the Government Eat Well Guide, which talks about eating vegetables, cereal foods, protein foods such as meat and vegetable alternatives, and dairy and alternatives. And the ideal way is for people to consume something from all of those. Ideally, people need to be reducing these discretionary foods such as the biscuits and cakes and crisps and so on, to make space for the more micronutrient-dense foods. Overall, we get calcium, magnesium from cereal foods, and actually you may not realise we get about 1/3 of our calcium from these foods in this country. We also get calcium, magnesium and protein from these dairy and alternatives. But also, you may also not realise we get quite a lot of calcium from our fruits and vegetables, particularly the vegetables, as well as magnesium and carotenes. And likewise, we get vitamin D also from the protein foods. We need to moderate our alcohol consumption in order to make sure our muscle particularly stay with us, I'm afraid. Bad news for people who like to drink a lot. Moderate is fine. Actually, we do have some work ongoing, which I haven't had a chance to show you today, which shows that it's particularly detrimental in women. So as well as a balanced diet, what else do we do? Well, we need to have enough vitamin D, which we can get from appropriate sun exposure, diet and supplements where necessary. And we really do need to keep active. We need to keep everyone moving, and we need population approaches to keep maintaining physical activity. And that can be walking, it can be doing whatever anyone can do within their capacity. It doesn't have to be Herculean. You don't have to do a marathon. You just keep moving. So a complete change of tack here. I'm nearly finished, and I'm going to talk a little bit about my career and how I got here. So this is a career path, and this is what we tend to think of as a career path. Time. You know, it's then and it's now, and you know, this is what I maybe thought this when I first started working, but actually the reality is rather more like this. A lot of moving around and changes. So I qualified in nutrition, dietetics, and I worked in the early days as senior dietitian in hospitals, and then moved to Somalia and Nigeria, and courtesy of my husband, in fact. And there I was doing nutrition surveys, and I measured body composition on over 2,000 adults and children over a period of 3 years. Then I came back and again worked in the NHS, and then I had my children, Douglas and Sarah, my twins, and clearly they weren't the size they are in this picture when they arrived, but clearly, you know, then one has to think about how you manage life, and so I did part-time work on government surveys, and then moved also to work on EPIC, which I've told you about, the validation studies for that. Then moved on to work on the Royal Society of Chemistry, Food Composition Database, McCann-Widderson, the composition of foods. And this is the data that underpins all that work I was showing you earlier. It's highly important that it's actually very well measured and so on. It's also the data that you see when you look at the packets, when you go shopping to see how much, you know, of something is in, how much nutrients, food, what's good, what's bad. It's all based on that data. And then I moved on to EPIC to work for much longer on that. For many, many years, probably longer than I care to think, and then came to UEA in 2007. So I was thinking about this and thinking, well, you know, this sort of, it's probably okay. It's not exactly a standard career path, but I was also thinking that over time it had given me some skills. So The clinical skills, I suppose, have really helped me to understand perhaps how to translate information and think about how we might translate what we're doing in policy and practice. I realised I'd been accumulating epidemiological skills over time, really, in many of the ways that I'd gone through, possibly, and in many ways I approached King's College London, who are now, about doing work, and I did this work of my own volition, but then that was also why I got the part-time work on these surveys and so on, and it went through. But also, having children is great, because one of the things it taught me was how to juggle, and anyone who's got a family will know what this means, trying to juggle time management and thinking, finding you've got a demand perhaps for some special thing to wear the next day, and you've got to put together a costume in time, and you've still got to get to work. So those are sort of things that one learns from this type of career, and gradually I suppose it has all come together. So you don't do epidemiology without large teams of people working together. Very briefly, to talk, I've already mentioned the PIs in EPIC, but this is, it was formed of a large number of people who worked either doing computing and data entry and data management, and the field workers in Norfolk and the study management team. This is quite a number of the EPIC Norfolk nutrition team who helped me build all those huge databases and computing systems that we've used and are still in use. And then Marlene and Angela, who also are continuing to work with me on publications and so on. And also we have the EPIC Europe colleagues too. But I think really we need to be thinking very hard about thanking the people who spend their time and energy giving to these studies. We wouldn't be able to do them without the people who contribute and have contributed time and time again over the years with not only reporting information but giving blood and so on. So really a final thank you too to colleagues, collaborators and family and friends, and particularly to thank my supervisor, Dr. Barbara Livingston and Katie Core, but also many of my UEA collaborators here, and I've listed a number of them, and I'm sorry if I've forgotten anyone. It wasn't deliberate, it's just I put together a list, particularly working with Diane Bunn, Jane Skinner, Lee Hooper, Richard Hayhoe, Amy Jennings, Donnie Cameron, Inish Mackers, and so on and so on. But also I wanted to thank PhD students who've done work in the past, current and past master students, and also the MBBS students who've been working with me on research. Again, also thanking other collaborators in EPIC Norfolk and collaborators across the UK, and some of who may well be watching in today on the streaming, and also those in the USA and the Far East. I haven't mentioned the collaborators in the UK Twin Study, Claire Steeves and Tim Spector, but also very great thanks to those. But particularly I want to thank my mentors, both formal and informal, who've supported me over time, and colleagues who, with their advice and discussions, have really helped me make decisions about my career and develop. And last but not least, I'd really much like to thank my family and friends and their tremendous support, and over the years, particularly when I've probably been rather boring being involved in some of the sort of work and working long hours. And particularly I'd like to thank my children, Douglas and Sarah, and my husband, David. So that's really all I'm going to see today, and I'm very happy to answer any questions if you have any.
Thank you very much, Elsa. I'm sure there will be some people in the audience who have burning questions. So, any questions from the audience, please? There's one over here.
Is the increase in interest in vegan diets likely to reduce the problems of aging?
Now, yes, now there's a sort of yes and no answer to that, and a very good one. Thank you, Marion.
So that was a question about vegan diet, and yeah. Is that going to increase problems in the future as summer with aging? Yes.
Right, so in terms of aging of muscle and bone, vegan eating is okay, providing people are very careful with what they do with their diet. So they need to make sure they're getting enough vitamin D from other sources. They need to make sure that they're being careful about their calcium intake, and they need to really be mixing their protein types to make sure that they have enough of the right protein, and that protein is sufficient to allow muscle growth. Overall, vegan eating patterns are actually very high in the types of food I was showing you today that are protective. So they're high in fruits and vegetables, and you know, they contain all those sort of wonderful compounds I was telling you about. So you know, they contain the micronutrients. They also contain a number of bioactive compounds, which I didn't have time to tell you today. I think overall, the other thing is that people with a vegan eating pattern very often have a much lower BMI, and compared with other people of the same age. And actually, we know having a high BMI, being overweight or very obese, is actually causing many of the problems that we have today in terms of onset of type 2 diabetes and cardiovascular disease. So I think the jury's out. We used to think it would protect against cancer and all sorts of things, but it's not certain yet.
Any other questions from the audience? Yes, there's a question here.
No, I know this first bit sounds slightly facile, but looking at the thing of micronutrient deficiency, it almost seems somebody eat plenty of broccoli and keep out of institutions. But more seriously, what can be done about problems of poor nutrition with people who are institutionalised?
Yes, and that's one of the things that we're addressing a lot more, and actually that's part of what the initiative I have, which I'm leading on the Optimising Nutrition and Hydration in Care Homes, is about. I didn't have time to mention this now, but I'm working with colleagues Lee Hooper and Diane Bunn, and then I know a lot of other organisations in the UK, so Age UK, the Care Home Caterers, we've got gerontologists and geriatricians, a lot of academic clinicians working in this area, and what we're trying to do is actually, what we found is there's plenty of guidelines, but actually there's much less in the way of knowing how well they're implemented and information so people can implement the guidelines properly, because often we have a very, very vague guideline. Well, it seems quite concrete, but actually when it boils down to it, for someone who doesn't know a great deal to actually interpret that to do something that makes a difference, it's difficult. So that's one of the things we're working on, and colleagues in that initiative are already working with improving the translation and the information that the Care Quality Commission use, for instance, for monitoring institutions and so on. But I think one of the things we need to think about more is malnutrition in the community, which is why I'm very interested in this area, because I think it's partly knowledge, but then there's also the issue of how we change policy and practice, and but unless we have the knowledge, we're not going to be able to do that.
Any other questions from the audience? There's one there.
It seems as an obvious deficiency in calcium, magnesium, and or proteins. One can get a habit from borrowing diet, or you can eat proper food. Is a real food really a much better way of getting the multivitamins than it is from the tablet?
Okay, that's a very good question too, and the question is, why don't you just take a supplement? Well, when you're asked to take a supplement medically, then you should, and they're very useful. Ideally, you prefer a dietary sources of these nutrients, partly because the molecular form in many supplements is not nature identical, and so that can cause problems. And some with the early supplementation studies, they found that actually the alpha-tocopherol beta-carotene trial actually increased rates of cancer, and some of that was because the early forms of those nutrients weren't identical. Ascorbic acid is that you get in a tablet is identical to what you would get in a vegetable or fruit, but some of the others also, the other issue with supplements is often the concentration. So you can get a magnesium supplement, but that's normally about 300 milligrams, and that's often enough to cause gut disturbances for people. Once you start to have a gut disturbance, you're less likely to be able to absorb your food in the first place, and you might be starting to counteract what you need to do. So ideally, if you can, then I think it's best to use foods, because also you're getting all the benefit of the other nutrients. For instance, fibre has finally hit the news again. We know that fibre is good for us, have a long time, but it's not been very media interesting until just recently again. But if you're eating a vegetable or a fruit or whole grain, you're getting a whole range of different nutrients and a lot of micronutrients as well. So ideally, where you can, it's best to use foods. If you can't, then obviously you have to have supplements and go with it.
There was a couple more questions.
Yes,
I'll take two. Well, there's one here. Yes.
Would you be able to comment on the increased prevalence of fracture rates in countries that consume a lot of dairy products?
Yes, actually at this stage I don't know the statistics, so I probably can't comment on that directly. Yes.
There's one over here. No? You've answered, it's been answered. Okay.
There's somebody up there as well, but if they've gone.
Hand gone down. Okay, well, Ilse, that was a tour de force. I might use the phrase a veritable feast for us this evening. And when I go home this evening, I know I've got a spag bol waiting for me. And I'm hoping there'll be lots of those colourful peppers in it. And I think I might add a slice of orange to my Aperol spritzer to ensure that no scurvy. I was fascinated to see your lifespan, and it ended at a peak at UEA. I hope that continues in an upward trajectory. Ladies and gentlemen, before we go out and look at the fare that's going to be presented in front of us and select some of those nutrients, could we please thank Ilse for a most My understanding is there's some nibbles and some wine, not to excess, ladies and gentlemen. Thank you. And I'm sure Ilse will answer further questions.